Have you ever wondered why our blood stops bleeding on its own so easily? What if it wasn't so easy? For people with blood clotting disorders like Haemophilia A, it's not as easy. Most people think that hemophilia patients die from large cuts and bleeding out, but actually they will end up dying and succumb to internal bleeding caused by the incapability of clotting. Hemophilia is a genetic disorder that can be a result of a missense and nonsense mutation which happen independently. Missense mutations involve the change in a single base pair which causes substitution of a different amino acid in the resulting protein. A nonsense mutation is a point mutation in a sequence of DNA that results in a premature stop codon. This will be transcribed into the mRNA. Usually a non-functional protein will be the product. Hemophilia A is an X-linked recessive mutation which causes a deficiency in the clotting factor 8. As you can see in this diagram, it is an example of X-linked recessive inheritance when the mother is the carrier of the condition. The mother will pass on her X chromosomes. In this case, the mutated chromosome is shown by a yellow X and the viable one is shown by a blue. Because it is a recessive gene, if a daughter receives one of the X chromosomes, she will not express the gene because she has another healthy one, which she received from her father. But if the mutated gene is passed on to a son, he will express the gene. Just because the daughter does not express the gene does not mean that it will not be passed on to her children. There's still a likelihood that it will be passed on to her children and they could possibly express it themselves. Families have their own distinct mutation. This is because hemophilia A can be a de novo mutation. This is the result of a mutation in the germ cell of a parent or in the fertilized egg itself. When tissues are injured, damage to a blood vessel may result in the leakage of blood through holes in the vessel wall. The vessels can break near the surface, as in a cut, or break deep inside the body, making a bruise. Platelets are small cells circulating in the blood. Each platelet is less than 100,000th of a centimeter in diameter. There are 150 to 400 billion platelets in a normal liter of blood. The platelets play an important role in stopping the bleeding by clumping together and forming a plug, thereby beginning the repair of an injured blood vessel. Here in the picture shown, the white cells beside your red blood cells represent your platelets, and the picture below represents them clotting and forming a fibrin clot around the damaged vessel. Clotting factors, like factor 8, are then needed to glue the plug in place, thus forming a clot. These proteins work like dominoes in a chain reaction called a convoluting cascade. Through these two diagrams, you can see the difference between normal clotting and hemophilia clotting. This is because at the top you can see the platelets fall like a domino effect, creating a clot over the wound. When looking at the hemophilia diagram, it is clear to see that the convoluting cascade stops and the clot is not able to form. When the protein is absent, the dominoes stop falling and the chain reaction is broken, as seen in the lower diagram. Clotting does not happen, or it happens at a much slower rate than normal. The platelets at the site of the injury do not mesh into place to form a clot, and the clot is easily displaced. The mutation is generated by the change in the base pair cytosine-guanine to thymine and guanine. On the sense strand, a nonsense mutation occurs. This makes the stop codon instead of the usual arginine amino acid. On the antisense strand, a missense mutation occurs. Usually, then, the arginine amino acid would then become glutamine amino acid, making factor 8 non-functional. Now, how does this happen? This change occurs by the methylation of cytosine 5', which is bonded to its base pair guanine, and subsequently the deamination of 5-methylcytosine to thymine, shown in the diagram. A hydrolysis reaction occurs, and then the deamination of 5-methylcytosine will follow, creating thymine. Without treatment, bleeding will continue until pressure outside the vessel is equal to the pressure outside. This can take days and sometimes weeks, unlike people who do not suffer from the mutation. Those who are diagnosed must wear a medical emergency bracelet to identify their condition. Currently, there is no cure for hemophilia, but scientists are always trying to find a cure. They believe the answer lies in gene therapy. Thank you for listening to my presentation. I hope it was enlightening.